Welcome to Cell Biology. Today, we are going to explore the fundamental building blocks of all living things. We will discover why a tiny bacterium and a complex human cell look so incredibly different on the inside. By the end of this lecture, you will not only know the biological differences, but you will understand the beautiful mathematics that actually forced these differences to exist. Let us dive in. Imagine you are looking at two very different types of houses. One is a simple, open-concept studio apartment, where the kitchen, bed, and living space are all in one single room. The other is a massive mansion with separate, walled-off rooms for cooking, sleeping, and working. In the world of biology, cells are exactly like these houses. They are divided into two main categories based entirely on their internal architecture. Understanding this architectural difference is the absolute foundation of all biology because it explains how life evolved from simple bacteria to complex human beings. Now that we have our analogy, let us formally define the simple studio apartments of the biological world: prokaryotic cells. A prokaryotic cell is a simple, single-celled organism that completely lacks a distinct, membrane-bound nucleus. It has no specialized internal compartments at all. The word itself comes from Greek, where pro means before, and karyon means nut or kernel, which refers to the nucleus. So these are the cells that existed before the nucleus evolved. The most classic, everyday examples of prokaryotic cells are bacteria. They are small, efficient, and incredibly resilient. Moving on from the simple studio apartments, let us look at the complex mansions, eukaryotic cells. A eukaryotic cell is a much larger, more complex cell that contains a true, membrane-bound nucleus to safely protect its DNA. Alongside this nucleus, it contains various specialized structures called organelles, which act like the separate rooms in our mansion. The prefix "eu" means true, so these are the cells with a true nucleus. Every plant, animal, and fungus you see around you, including your own body, is made entirely of these highly organized eukaryotic cells. You might be wondering why we care so much about how a cell is organized inside. This distinction is actually a matter of life and death in modern medicine. When you get sick with a bacterial infection, doctors prescribe antibiotics. These drugs are specifically designed to attack the unique structures of prokaryotic cells, like their special cell walls. Because your own body is made entirely of eukaryotic cells, the medicine destroys the invading bacteria without harming your own tissues. Furthermore, this structural divide helps scientists trace the evolutionary history of life on Earth. Now that we understand what these two cell types are, we must ask a deeper question. Why are prokaryotic cells so small and simple, while eukaryotic cells are large and complex? To truly understand this, we have to look at the mathematics of cell size. The most critical mathematical concept in cell biology is the surface area to volume ratio. This single ratio dictates exactly how big a cell can get before it literally starves to death. It is a strict biological rule of life governed entirely by geometry. This geometric rule leads us to a beautiful and simple formula. If we assume our cell is a perfect sphere, the surface area to volume ratio, denoted by R, simplifies to 3 divided by the radius, r. Conceptually, this formula tells us how much outer membrane is available to feed the living cytoplasm inside. The variable r is the ratio itself, and r is the radius of the cell. Notice that r is inversely proportional to r. This means that as the radius of the cell increases, its surface area to volume ratio strictly decreases. A larger cell has much less surface area relative to its massive volume. Before we move on, let us see this formula in action with a concrete example. Let us calculate the ratio for a typical small prokaryotic cell, like a bacterium. We are given that this bacterial cell is roughly spherical with a very small radius of 1 micrometer. We want to find out exactly how much surface area it has to support its internal volume. We choose our simplified ratio formula and plug in the value 1 for the radius. 3 divided by 1 gives us a final ratio of 3 inverse micrometers. Physically, this high number means the bacterium has a massive amount of outer membrane compared to its tiny internal space, allowing rapid feeding through simple diffusion. Now that we have calculated the ratio for a tiny bacterium, let us look at a typical eukaryotic cell, like one of your own skin cells. These cells are much larger. We are given a radius of 10 micrometers, and we need to calculate its surface area to volume ratio. We use the exact same mathematical approach, substituting 10 for the radius. 3 divided by 10 gives us a final answer of 0.3 inverse micrometers. This number is 10 times smaller than the ratio we found for the tiny prokaryotic cell. Physically, this means the large eukaryotic cell has very little outer membrane to support a gigantic internal volume, creating a severe physical crisis for survival. Now that we have used this formula to see the crisis large cells face, let us understand where it comes from. We are going to derive the simplified formula we just used. We start with the fundamental definition. The ratio R equals the total surface area divided by the total volume. To make this specific to our spherical cells, we need the geometric formulas for a sphere. The surface area of a sphere is 4 times pi times the radius squared. The volume of a sphere is 4/3 times pi times the radius cubed. In our next step, we will combine these pieces to see how the mathematics naturally simplifies. Continuing our derivation, we substitute the formulas for surface area and volume into our main ratio equation. The numerator is 4 pi r squared, and the denominator is 4/3 pi r cubed. Now we look for common terms to cancel. The 4 and the pi in the numerator and denominator cancel out perfectly. We also divide r squared by r cubed, which leaves a single r in the denominator. When we simplify the remaining fraction of 1 divided by 1/3 r, the 3 flips to the top. We are left with the beautiful and simple result. The ratio equals 3 divided by the radius. This proves exactly why larger cells mathematically lose surface area. So we have proven that as a cell gets bigger, its surface area to volume ratio plummets. This brings us to a critical biological problem. Because the ratio for a large eukaryotic cell is so low, nutrients cannot simply diffuse from the outside to the center fast enough to keep the cell alive. The cell would literally starve. This mathematical limitation is exactly why eukaryotic cells were forced by evolution to develop internal membrane-bound organelles. Structures like the endoplasmic reticulum act as an internal highway system for transport, completely bypassing the limitations of simple diffusion. Let us bring all of this together and look at exactly what we have been discussing. On the screen is a side-by-side comparison of our two cell types. On the left, you see the tiny prokaryotic cell, the bacterium. Notice its simple interior. There is no nucleus, just a tangled region of DNA, and it relies on its outer capsule and cell wall. Now, look at the right side. This is the massive eukaryotic cell. Right in the center is the large protective nucleus. Surrounding it is a complex maze of organelles, like the folded endoplasmic reticulum and the mitochondria. The scale bar at the bottom perfectly illustrates our mathematical lesson. The eukaryotic cell is vastly larger, requiring this complex internal city to survive. We have covered a lot of ground today, so let us summarize the most important ideas. First, remember the fundamental structural divide. Prokaryotic cells are small and simple, without a nucleus, while eukaryotic cells are large and complex, with specialized organelles. Second, we learned that this difference is not random. It is driven by the mathematics of the surface area to volume ratio. Finally, we saw that the complex organelles in eukaryotic cells are an evolutionary solution to the physical crisis of being large. You now hold the key to understanding how all complex life is built on a microscopic level. Excellent work today.